Good afternoon, everyone. Today, Jared Gazal and I will be presenting our BIPN 105 student project on the physiological regulation of fluid balance and capillary exchange. Throughout this presentation, we will present data showing the changes in the weight of a frog, as well as variations in the drip rate of fluid from the frog's toes in response to perfusion of the frog aorta with ringer/dextran solutions that contain various drugs. Specifically, we assessed how introducing isoprenolol, epinephrine, and prazosin into the perfusate altered overall fluid movement. As for the overall objective of this experiment, by using these particular drugs, we want to uncover the impact of different types of adrenergic agents on fluid movement and capillary exchange.
As you all know, in order to obtain the data, we had to insert a catheter into the frog's aorta to allow for fluid perfusion. The Ringer's and dextran solution was used to maintain osmotic balance in the control conditions, and the controls were recorded prior to the addition of any other drugs. During the experiment, we focused on monitoring weight change to assess fluid movement into our in or out of tissues, as well as drip rate, which was utilized as an indicator of blood flow and vascular resistance. To actually analyze this data, we were able to use Starling's equation to explain the change in weight due to fluid filtration and reabsorption, while Ohm's law was used to understand changes in drip rate in response to changes in vascular resistance.
Calibration of the isometric force transducer was necessary to be able to convert the transducer output, which was in millivolts, into a weight in grams. The conversion factor here was 0.185 grams per millivolt to convert a slope into change in weight in grams per minute, which Ghazal will explain next.
The voltage data from the transducer was used to determine the rate of weight change in the frog through a linear regression analysis. Applying the linear regression analysis to the raw transducer voltage measurements, which is in millivolts per second, yielded a slope value that represented the rate of change in voltage in millivolts over time in seconds. By multiplying the obtained slope of the linear regression by the conversion factor obtained in the figure, the rate of weight change in grams per second was calculated. Under controlled conditions, the frog exhibited a weight change of 0.0804 grams per minute and a drip rate of 18 drips per minute. After the introduction of the isoproterenol, both the weight change increased to 0.1104 grams per minute and the drip rate increased to 26 drips per minute. This indicates a trend of increased fluid movement compared to the controlled conditions. Isoproterenol is a beta-1 and beta-2 adrenergic receptor agonist. While the activation of beta-1 receptor affects the cardiac functions, beta-2 adrenergic receptors are located mostly on the smooth muscles and the vascular, and their activation leads to the vasodilation. In this experiment, an increase in the weight change and drip rate following perfusion with the Ringer's solution containing isoproterenol was observed. This suggests that isoproterenol activates the beta-2 adrenergic receptor, and the beta-2 adrenergic receptors are G proteins coated with receptors located on the arterial smooth muscle cells. When isoproterenol binds to these receptors, G protein signaling cascade is activated. Once the activated G proteins activates the adenylyl cascade, which is AC, which catalyzes the conversion of ATP to cyclic AMP. The accumulation of the intercellular cyclic AMP results in activation of protein kinases A, which is pKa, which phosphorylates myosin light chain kinases, which is MLCK, effectively inactivating it. MLCK is responsible for phosphorylating myosin light chain, which is MLC, to promote the actin-myosin cross bridge formation that is necessary for contraction initiation. As a result, the inhibition of the MLCK reduces myosin phosphorylation and prevents contraction. In addition, MLCK inhibition and the pKa mediated phosphorylation leads to an activation of potassium channels. Lastly, the potassium influx due to the activation of these channels results in the hyperpolarization of the smooth muscle cells, which further promotes relaxation. Together, these effects contribute to the atrial smooth muscle relaxation and then vasodilation, which increases weight change and drip rate.
Right, for epinephrine, the control condition showed a weight change of 0.0238 grams per minute and a drip rate of 20 drips per minute. With the addition of epinephrine, the weight change became negative, indicating fluid loss, and the drip rate decreased to 12 drips per minute. This suggests a shift from fluid accumulation to fluid reabsorption. In terms of mechanics, epinephrine is an alpha-1 and beta-2 adrenergic receptor agonist. While alpha-1 activation leads to vasoconstriction, beta-2 activation promotes vasodilation. In this experiment, the decrease in the rate of weight change and drip rate following perfusion with Ringer/dextran solution containing epinephrine suggests that vasoconstriction predominated. This outcome indicates that epinephrine preferentially activated alpha-1 receptors in the arterioles. Alpha-1 receptors are GQ protein coupled receptors located on the arterial small smooth muscles. When these receptors are activated by epinephrine, the GQ protein signaling the cascade of events is triggered. To begin, the GQ protein activates phospholipase. C. It's an enzyme that cleaves a PIPes, another thing called PIP2 into two secondary messengers, IP3 and DAG. IP3 binds to receptors on the sarcoplasmic reticulum, triggering the release of calcium into the cytoplasm. DAG, on the other hand, activates protein kinase C, enhancing calcium sensibility in contractile proteins and delaying repolarization through potassium channel inhibition. This increased intracellular calcium binds to calmodulin. This forces a calcium-calmodulin complex that activates myosin light chain kinase. Myosin light chain kinase then phosphorylates myosin light chains. This enables actin-myosin cross-bridge formation, which are necessary for smooth muscle contraction. This smooth muscle contraction of the arterial walls then manifests as vasoconstriction of the arterioles.
We also looked at the effects of prazosin by itself as well as in combination with epinephrine. With prazosin alone, we saw the weight change increase slightly and then increase even further when epinephrine was also present in the perfusate, showing a gradual increase in both fluid accumulation and drip rate across the treatments. Prazosin affects as an alpha-1 adrenergic receptor antagonist. So it does this through a competitive blockade, meaning that prazosin can compete with alpha-1 adrenergic receptor agonists like epinephrine in binding with the alpha-1 receptors. Under normal circumstances, alpha-1 adrenergic receptors are GQ protein coupled receptors that trigger that pathway that was discussed earlier. However, unlike epinephrine, binding of prazosin to alpha-1 adrenergic receptors does not activate the signaling cascade. As a result, the addition of prazosin alone would not be expected to alter vascular resistance. So the slight increase in weight change and drip rate that we observed is likely due to some epinephrine from the previous treatment not being fully cleared from the circulation. Once we did add epinephrine into the perfusate, we did see a more stark increase in the weight change. This is because the prazosin is blocking the alpha-1 receptors, and so epinephrine can no longer bind to the alpha-1 receptors. However, epinephrine is also able to bind to beta-1 receptors, which are associated with vasodilation. So it kind of unmasks the ability of epinephrine to bind to the beta-1 receptors, which ultimately leads to a vasodilation.
Adrenergic agents like isoproterenol and epinephrine significantly influence vascular resistance and fluid dynamics. Activation of beta-adrenergic receptors leads to a vasodilation and increases capillary hydrostatic pressure, enhancing filtration, while alpha-adrenergic receptor activation induces vasoconstriction and reduces fluid movement. For the isoproterenol treats heart blocks and asthma by enhancing cardiac output and bronchodilation. Epinephrine is crucial for Anaphylaxis, counteracting severe bronchial constriction from histamine release, while prazosin lowers hypertension by increasing blood muscle radius and reducing resistance, and improving blood flow per Ohm's law and Starling's equation. Thank you for listening.